Hi guys, this is the final video of the cardiovascular system, and in this video we're going to be looking at tissue flow and capillary exchange. With regard to blood flow to specific organs or tissues, there are local or intrinsic determinants of flow based on the needs of that tissue. What we will be looking at is local controls over the pressure within vessels that supply specific areas of the body that override what is happening with pressure in the rest of the body. Put another way, this system works independent of the extrinsic mechanisms that control MAP throughout the body. And that's why these mechanisms are called autoregulation. Blood flow through tissues is known as perfusion, and perfusion must meet the needs of the tissues. Otherwise, there will be too much or too little blood going to an organ. Flow slows way down at the tissue level to allow time for exchange, and we will be looking at exchange as the last topic for this video. Flow is controlled through resistance, and resistance is controlled by the diameter of the arterial blood vessels supplying an organ. In this picture, we see an example of blood being routed away from the lower extremities through vasoconstriction and routed toward the digestive organs through vasodilation after eating a meal. And the opposite is occurring when blood is needed in the lower extremities, especially the skeletal muscles. Not only can blood be rerouted from one organ to another, but blood can also be rerouted from one tissue area within an organ to another. And that's how fine-tuned this system is. So how is this accomplished? When a tissue is not adequately perfused, there is a buildup of metabolic waste products, and this will initiate local responses to increase blood flow. Some of these responses include the release of paracrines, which act as local hormones, such as nitric oxide, which is a powerful vasodilator, and endothelin, which is known to be the most potent vasoconstrictor. During trauma or immune response, the release of inflammatory chemicals will cause vasodilation. These are some of the metabolic controls for the autoregulation of blood flow. Blood vessels will also respond to a passive stretch caused by pressure to the wall of the vessel, and this causes a reflexive vasoconstriction. And the opposite is true as well, that a reduced stretch would cause vasodilation. And this response of a vessel to its own input is known as a myogenic control. In the event that an organ develops an increased need for blood over an extended period of time, new blood vessels will form and grow to supply the increased demand, and this is known as angiogenesis, and it's considered a long-term regulation of blood flow. This picture is a great synopsis of both the intrinsic and extrinsic controls of the arterial smooth muscles. If you read the caption of this picture in the textbook, you will better understand how the sympathetic nervous system can increase blood flow to certain organs and at the same time decrease blood flow to others. Let's now turn our attention to what happens as blood enters the capillary bed, and we'll spend the rest of the video talking about that topic. But before we go there, we must review some basic concepts regarding osmosis. Recall from A&P1 our experiment with a semi-permeable membrane and some dissolved solutes. These dissolved solutes are non-penetrating. They're too large to move through the membrane. And the solvent in our case is water. This container is broken up into two compartments by a semi-permeable membrane, and there's an equal volume of water on either side. To the left side, we add a large non-penetrating solute, such as albumin, and now the concentrations are different. So side A is more concentrated than side B. We give the system some time to equilibrate, and what we find is that the water moves from side B to side A, and this equalizes the concentrations on both sides. The pull of water to the left by a more highly concentrated solution is known as osmotic pressure, and the solution that has a higher concentration is said to have a higher osmolarity because it has a greater attraction for water. Now the weight of the water on the left has an opposite effect on the movement of water, and this is known as hydrostatic pressure. Hydrostatic pressure builds because of the unequal volume of water. And when these two forces are equal, there's a net zero movement. And that just means water is passing equally from both sides. And this is the equilibrium point. This picture from your textbook is showing the same analogy. However, in the picture on the left, it's showing a barrier pushing the water to the right. This barrier or piston is mimicking the effects of hydrostatic pressure from an external influence, such as blood pressure. And so these are the forces that will drive fluid flow and exchange at the capillary level. So let's take a look at that now. The first thing I'm going to do is tell you the ending of the story. Under normal physiological conditions, at the arterial end, the conditions favor filtration, and at the venial end, the conditions favor reabsorption. Here are the numbers that I would like you to commit to memory. In the arterial end of the capillary, the blood hydrostatic pressure is 35 millimeters of mercury, and at the venial end, it is 17 millimeters of mercury. None of the other numbers are important for you at this time. What is most important is whether or not the other forces driving fluid movement can overcome the hydrostatic pressure. So let's look at what those other forces are. First, we have the hydrostatic pressure, and wherever we have fluid, we will have hydrostatic pressure. Within the vessel, it is the same thing as the blood pressure, and it will have a tendency to push the fluid out of the vessel. Any fluid outside of the vessel, but not within tissue cells, is known as interstitial hydrostatic pressure. And this pressure would tend to push the fluid back into the vessel, opposing the blood hydrostatic pressure. There tends to be minimal fluid in the tissues, so this number is usually very small or even a negative number, denoting a slight vacuum effect. At the capillary level, most fluids and small dissolved substances will be leaving the capillary at the arterial end. What is left behind in the vessel are the formed elements of the blood and the larger proteins. And these proteins tend to have an osmotic pull on water because of their negative charges. And this force is called colloid osmotic pressure. Because these proteins do not leave the vessels, there tends to be a much higher colloid osmotic pressure within the vessel than outside. And it tends to be very stable from arterial end to venial end. Let's take one more look at our capillary picture to describe these forces that determine the net flow of fluid. And please do not worry if these numbers do not match the numbers from your textbook. As I've said many times in class, there are so many different variables when determining these physiological values that you will rarely have agreement on an absolute value. And what is most important is the physiological process that is taking place. As blood enters the capillary from a met arteriole, the blood hydrostatic pressure is around 35 millimeters of mercury, and the colloid osmotic pressure within the blood is lower than this. And we know that the interstitial hydrostatic pressure and the interstitial colloid osmotic pressure are both minimal. So what will determine the direction of fluid movement is basically the blood hydrostatic pressure, and that it does. And at this end, we say that conditions favor filtration, and a major portion of the blood fluid leaves as serum at this end of the capillary, taking it with it dissolved oxygen and nutrients and other necessary substances that are needed by the nearby tissue cells. As the blood continues to move through the capillary, much more concentrated now, it has lost about 50% of the pressure behind it, and by the time it reaches the postcapillary venule, it's at around 17 millimeters of mercury. At this point, the blood hydrostatic pressure has become less than the blood colloid osmotic pressure, and the porous capillary and postcapillary venules drink up this fluid like a couple of thirsty sailors. Along with this fluid comes carbon dioxide and other waste products that the tissue cells have dumped out onto their sidewalks, sort of what my block looks like on a Tuesday morning before the garbage crew comes. At this end, we say that conditions favor reabsorption. Any fluid that does not get reabsorbed by the postcapillary venule gets picked up by lymphatic capillaries that are embedded throughout these capillary beds. And as this picture from your textbook shows, an astounding 20 liters of fluid gets filtered from the capillaries each day, 17 liters of which will be picked up at the venous end, and 3 liters of which will be picked up by the lymphatic capillaries. Aren't our bodies just amazing? What an incredible system that works so well that you have no clue that this is going on a continual basis until something throws off the system. And thankfully, we have well-educated doctors and nurses who know how to deal with such imbalances, who can help us get back on the right track to restore homeostasis. Well, that's it, guys. That's the end of the material for Exam 1, and I'll see you soon.